In 1961, Marshall Nirenberg and his graduate student Heinrich Mattaei conducted an experiment to attempt to figure out how genetic code is translated. In order to be able to control the experiment, the pair needed a cell-free system. This system is created when the cell walls are ruptured, releasing cytoplasm to be synthesized with RNA. Using a mortar and pestle, they grounded up E. coli bacteria cells to collect the cytoplasm for their experiment. 20 test tubes were used and filled with the different amino acids. In each test tube, 19 of the amino acids were unlabeled and considered cold, but one amino acid was labeled and considered hot because it was radioactive. By changing the amino acid that was radioactive, they could see which one would be incorporated into a protein when a particular type of synthetic RNA was added. Their major discovery was when they used synthetic RNA from uracil, and the hot test tube contained radioactive phenylalanine. The hot test tube contained thousands of counts per milligram of the protein, whereas the 19 cold test tubes contained approximately 70 counts each. This meant that a chain of uracil bases was creating the protein phenylalanine. They also discovered the codons for lysine and proline in this experiment by using the same technique. In 1964, Marshall Nirenberg and Philippe Ledier found a new method to speed up the process of deciphering the genetic code. Nirenberg could not use the first method he has previously used with Mattaei because mRNA bases were being taken up randomly by ribosomes, making it difficult to correlate an amino acid with a codon. So instead, they used short RNA sequences in cell-free systems. The known RNA sequences were mixed with ribosomes and bound with the tRNA molecule that was complementary to the codon. Each amino acid was labeled before being put through a millipore filter. This filter would allow unbound tRNAs to pass through, but would catch any ribosomes that were bound with the triplet. The caught ribosomes were then tested for radioactivity, and if they were radioactive, then they were corresponded with the labeled amino acid. Using this method, Nirenberg and Ledier identified the remaining 61 codons and the amino acid they coded for. Thanks for watching.